Grade 10, today we're going to talk about cellular respiration. Check your book, chapter 4, section 5. So cellular respiration, we're going to start by glycolysis. As we said before, lysis means breaking down. So glycolysis means the breaking down of glucose. Glycolysis is an ongoing process in all the cells, including yours. It takes place in the cytoplasm before cellular respiration and it does not require oxygen. Glycolysis makes a small number of ATP molecules, but its other products are much more important. If oxygen is available, the products of glycolysis are used to produce many more ATP molecules through cellular respiration. So glycolysis is going to be summarized as follows. First of all, we're going to look over here. We have two ATP molecules that are used to energize a glucose molecule. The glucose is split into 3 carbon molecules. We said it is called pyruvate. Pyruvate, each one of the molecules of pyruvate contains 3 carbons. Then a series of enzymes and chemical reactions are going to rearrange these 3 carbon molecules. The energized electrons, here they are, the energized electrons from the 3 carbon molecules are going to be transferred to NAD, nicotinamide adenine dinucleotide. Molecules of NADH are formed. And then a series of reactions are going to convert the 3 carbon molecules to pyruvate, which enters cellular respiration. Here we have 4 ATP are formed. But instead of, despite the fact that 4 ATPs are formed, we only get 2 ADP as a result, because the other 2 ADPs were used over here to break down glucose. So glucose broken down, the presence of 2 ATP producing 2 molecules of pyruvate, and then producing also NADH, which is very important. So although glycolysis makes 4 ATP molecules, 2 ATPs were used in the first place to split the glucose molecule. So the breakdown of 1 glucose molecule by glycolysis gives a net gain of 2 ATPs. The pyruvate and NADH produced by glycolysis are used for cellular respiration when oxygen is present. NADH, of course, is an oxygen carrier. Then we go to the Krebs cycle. The Krebs cycle, we're going to go through it slow, step by step, all what you need to know in the Krebs cycle. So, in the Krebs cycle, first of all, we have the pyruvate, which was formed, is going to be broken down. The pyruvate is going to be split into 2 carbon molecule and a molecule of carbon dioxide. The pyruvate, which was formed, is broken down into a 2 carbon molecule and a molecule of carbon dioxide, which is given off as a waste product. High energy electrons are transferred from the 2 carbon molecule over there into NAD, forming NADH. The NADH is going to move to the electron transport chain. So first of all, pyruvate broken down into a 2 carbon molecule and carbon dioxide. This carbon dioxide is leaving. This 2 carbon is going to help the NAD transformed into NADH, which is an electron carrier. Then what do we have next? The coenzyme A. Here is the coenzyme A. The coenzyme A is going to bond the 2 carbon molecules formed from the breakdown of pyruvate. And then this intermediate molecule is going to go to the Krebs cycle. What do we get here? Pyruvate broken down, carbon dioxide leaving, NAD transformed into NADH, first part of the Krebs cycle. Then coenzyme A is going to help forming this intermediate molecule, which is called acetyl-CoA, and it's going to be part of the Krebs cycle. The Krebs cycle is going to lead to the formation. At this point, what do we get? We get citric acid. This is why the Krebs cycle is also called the citric acid cycle. So the 2 carbon part of the intermediate molecule is added to a 4 carbon molecule to form a 6 molecule citric acid. Here is the citric acid which is formed of 6 carbons. As soon as it's formed, it is going to be broken down. So the breaking down of the citric acid is going to happen by an enzyme, and it is going to give us a 5 carbon molecule. Count them down: 1, 2, 3, 4, 5. So citric acid is formed, then citric acid broken down to give a 5 carbon molecule. After the formation of this 5 carbon molecule, a molecule of NADH is made, and it's going to move out of the Krebs cycle, and a molecule of carbon dioxide is giving out as a waste product. Pyruvate broken down, we're going to get a 2 carbon molecule. We're going to get coenzyme A. Coenzyme A is going to help the formation of citric acid. Then citric acid broken down, yielding a 5 carbon molecule. NADH is going to leave the Krebs cycle, and carbon dioxide is leaving as waste product. Then this 5 carbon molecule is going to be broken down. So the breakdown of this 5 carbon molecule is also going to happen in the presence of an enzyme. We're going to get a 4 carbon molecule. Here is it: 1, 2, 3, 4. A 4 carbon molecule. We're going to get it. We're going to get NADH, and we're going to get a molecule of ATP. So at this point, we're breaking down 6 into 5, and then 5 into 4. Here we get NADH that leaves, and we get carbon dioxide. Here we're going to get the 4 carbon molecule, and we're also going to get NADH. But also a molecule of ATP is formed. NADH is going to also leave the Krebs cycle, and the carbon dioxide is given off as a waste product. These 4 carbons are going to be rearranged. The rearrangement of these 4 carbons is going to give out high energy electrons. These high energy electrons, how do we see them? We see them as fat H2, and we see them as NADH. Fat H2 is another electron carrier. So we get more electron carriers, and it happens through the rearrangement of these 4 carbon molecules. You don't need to know the names of the 4 carbon molecules. We're demonstrating them here so you can see the steps. So the 6 carbons broken down giving 5. The 5 carbon broken down giving 4. Here we get NADH. Here we get NADH. Don't forget that NADH are electron carriers. Electron carriers are very important. We're going to say why in a few seconds. Here the 4 carbons are going to rearrange to give us more electron carriers like fat H2, but at the same time we're going to get ATP. So what remains here? We're going to have the 4 carbons at the end. The products of the breakdown of 1 molecule of pyruvate are 3 molecules of carbon dioxide. Here they are, 3 carbons of carbon dioxide that are going to be given off as waste products. 1 molecule of ATP, 4 molecules of NADH, and 1 molecule of fat H2 to the electron transport chain. So glycolysis produces 2 pyruvate molecules. Therefore, these products are half of what comes out for 1 glucose molecule. The total are for the breakdown of 1 glucose molecule: 6 carbon dioxide, 2 ATP, 8 NADH, and 2 fat H2. So the electron transport chain is, why are we talking about electron transportation as if this was the most important part? Because it is. The electron transport chain takes place in and across the inner membrane of the mitochondrion. As with the electron transport in photosynthesis, the proteins make up the electron transport chain in cellular respiration. The proteins are going to use the energy from the electrons supplied by the NADH and fat H2 to pump the hydrogen ions against a concentration gradient. Remember when we said that everything is going to move down the concentration gradient if it is passive transport, but in case of active transport it is going to require energy. So in order to push the hydrogen ions against a concentration gradient, the electrons are going to be supplied by this NADH and the FADH2 in order for them to pass in and across the inner mitochondrial membrane. The ions are going to flow back later through the membrane to produce ATP. The oxygen is needed at the end of the process to pick up the electrons that have gone through the chain. After the production of NADH and FADH2 and the production of ATP, multiple more steps are going to happen. First of all, the electrons are going to be removed. The cell needs these electrons. The electrons are removed. The proteins inside the inner membrane of the mitochondrion are going to take the high energy electrons from where? They are going to take them from the NADH and the FADH2 to where 2 molecules of NADH and 1 molecule of FADH2 are used. So first of all, the electrons are going to be used. Why are we building all of these high energy electrons carrier? In order for the membrane to carry them. Why does the membrane need them? It needs them to pump the hydrogen. Look here, the hydrogen is pumped. Why do we call it pumping? Because this hydrogen is going to move against the concentration gradient. It needs energy. This is why we need the high energy carriers. They are carrying electrons. The NADH and the FADH2 are carrying the electrons from food. So the hydrogen ions are going to be transported. The high energy electrons, they are going to travel through the protein in the electron transport chains. The proteins use the energy from the electrons to pump the hydrogen ion. Here they are. The hydrogen ion, they are going to be pumped across the inner membrane to produce a chemiosmotic agent. Chemiosmotic agent, this is an agent that is going to move against the concentration gradient. Remember when we talked about osmosis, we said that this is the movement of the water across the cell membrane down the concentration gradient. But in the case of chemiosmosis, it is going to be the movement of ions across the membrane of the mitochondrion down the electrochemical gradient. Where does this come from? It came from the presence of the ATP and the movement of hydrogen. So again, chemiosmosis is going to happen here. It is going to push the hydrogen, but it is going to be down the electrochemical gradient. It is going to create a difference in electricity from here to there. So the hydrogen ions are going to be transported. The proteins are going to use the energy from the electrons to pump the hydrogen ions across the inner membrane, and the hydrogen ions are going to build up inside the inner mitochondrial membrane. What do we get next? We get ATP. At this point, just as in photosynthesis, the flow of hydrogen ion is used to make ATP. The hydrogen ions are going to diffuse through a protein channel. Here is the protein channel. They're going to diffuse through the protein channel in the inner membrane of the mitochondrion. The channel is part of the ATP synthase enzyme. ATP synthase. Synthesis means making. So ATP synthase is an enzyme that produces ATP. ATP synthase is going to add the phosphate group to ADP to make ATP. Each pair of electrons that pass through the electron transport chain, an average of 3 ATP molecules are made. Then finally, what do we get? Water is formed. Oxygen is going to finally enter the cellular respiration process. At this point, the oxygen is going to pick up the electrons over here. This is where oxygen is going to enter the cellular respiration. So at this point, the oxygen is going to enter and it's going to pick up the electrons and the hydrogen ions to form water. The water is giving off as a waste product. So here what happened? First of all, the electrons were removed. The proteins inside the inner membrane of the mitochondrion, they are going to take the high energy electrons from the NADH and the FADH2, and then the high energy electrons, they are going to travel through the proteins in the electron transport chain. The proteins, they are going to use the energy from the electrons to pump the hydrogen ions. They are going to pump the hydrogen ions to produce a chemiosmotic gradient. They are going to cause a difference in concentration gradient between the inside of the membrane and the outside of the membrane, the inner membrane and the outer membrane of the mitochondria. Okay, so in this, the hydrogen ions are going to build up where? On the inside of the inner mitochondrial membrane. Then this flow of hydrogen is going to lead to the formation of ATP. So NADH and FADH2 and the flow of electrons is going to help the pumping of hydrogen, creating a chemiosmotic gradient, which is a concentration gradient that allows the hydrogen ions to be concentrated along the inner mitochondrial membrane, leading to the formation of ATP. ATP produced by the presence of ATP synthase, which is going to add 1-phosphate to the ADP, leading to the formation of ATP. And then water is formed. Water, in the presence of the oxygen, this is where the oxygen is going to enter the cellular respiration. Oxygen, it is going to enter the cellular respiration, it is going to pick up the electrons and the hydrogen ions to form water, and then water is released as a waste product. So the products of the cellular respiration, including glycolysis, are carbon dioxide from the Krebs cycle, water from the electron transport chain, and what we need, 38 ATP molecules for every glucose molecule. 2 from glycolysis, 2 from Krebs cycle, and 34 from the electron transport chain. This is what your cells needed. Your cells needed a lot of ATPs in order to perform their growth and to perform their repair and to perform all of their functions. So this is a wrap up of the cellular respiration. Next session, I'm going to make a summary for cellular respiration and for all of the Krebs cycle in order for you to have them in steps, and then we're going to start the fermentation. Thank you very much.